Hi, I'm Bill Kasman, and I'm the founding director of GeneWorx Capital, and welcome to another edition of the GeneWorx Discovery Series, where we feature science, people, and ideas from the leading edge of life science. In this month's live event, we're featuring a company that's developing groundbreaking technology to accelerate the fight against cancer. According to the American Cancer Society, over 600,000 people die from cancer in the US each year. That's 600,000 people each year just in the US. That's an urgent situation. There are 126 approved cancer medicines, but clearly they don't work well enough for all cancers. On average, it historically takes 8 years to develop and approve new cancer medicines, and 97% of those in clinical development fail to receive FDA approval. Biological engineering is transforming this. Let's compare it to COVID-19. Prior to 2020, the average development time for a new vaccine was 10 to 15 years. On December 31st of last year, the outbreak of COVID-19 in Wuhan, China was first reported. On January 11th, China released the fully sequenced SARS-CoV-2 genome to scientists around the world. And just 42 days later, the first vaccine was on its way to the National Institute of Allergy and Infectious Diseases in Bethesda, Maryland, to begin human testing. As of today, there are 54 vaccines in human clinical trials, plus another 87 in preclinical animal studies. Widespread vaccinations of the population are poised to commence in under a year from the original COVID-19 outbreak. Biological engineering breakthroughs have empowered scientists with the knowledge and tools to achieve this astounding feat. Motivated by an urgent crisis, the vaccine development process has been completely transformed, saving millions of lives from COVID-19 and other diseases in the future. 600 cancer deaths per year just in the US is also an urgent crisis. Now, let's imagine if we had a research platform that could accurately model the most intractable human cancers and predict the response to new therapies before we ever begin clinical trials in humans. How might we drive down that 97% new drug failure rate, shorten the 8-year development time, accelerate new cures, and save millions of lives? Well, today we're going to hear from a company that has unleashed the latest biological engineering tools on this challenge. GeneWorx is really excited to introduce you to Dr. Adrienne Watson, Vice President of R&D at Surrogen and an expert in gene editing technology for swine models of human diseases with a focus on cancer, neurodegenerative diseases, and regenerative medicine. She's led a team at Surrogen to create the world's first engineered immune-competent large animal model of glioblastoma, rapidly induced genetically defined liver tumors nearly identical to human hepatocellular carcinoma, and many others. Dr. Watson will speak for approximately 45 minutes, and then we'll have 15 minutes for questions. Put your questions into the Q&A as we go, and I'll serve them up to Adrienne at the end. So with that, I'll turn it over to Dr. Watson.
Well, thank you, Bill. In a world that's demanding faster and more innovative healthcare solutions, I'm really excited to tell you all today about how we at Recombinetics and Surrogen are harnessing the power of the pig in pursuit of novel cancer therapies. I'll start with a little bit of background on our company. So the parent company to Surrogen is Recombinetics, and Recombinetics achieves its mission through 4 divisions. Today, I'll be telling you about Surrogen, which is our preclinical research division, which is responsible for biomedical models to develop human therapeutics. We also have 2 regenerative medicine subsidiaries called Regenavita and Mecana, as well as a food animal agricultural division called Excelligen, which I will not be talking about today. But the parent company, Recombinetics, was founded in 2008, and we completed a $34 million Series A capital raise in August of 2018, led by a strategic investor from healthcare, which has really led us to focus a lot over the past few years on our biomedical and human applications. Recombinetics has 10 PhD scientists and 4 DVMs with over 65 years of experience in modeling diseases in large animals, with a lot of experience, particularly around the oncology and cardiovascular fields. We also combined have over 100 years of genome engineering experience, and a lot of the work that we've done to date has been funded through grants. So we've had 33 grants totaling over $25 million from NIH, DoD, USDA, and other nonprofits. We have a large patent portfolio with 68 patents issued and 192 patents pending and licensed under site-specific nuclease patent rights. So Surgeon, which is focused on developing biomedical models for preclinical development, was really came from the fact that, as Bill mentioned, clinical development fails at a very, very high rate. And so if you look at the cost and time for a new drug to be developed, it costs nearly $3 billion for a new drug to go from preclinical studies through FDA approval, and often takes up to 15 years. It takes thousands and thousands of patients for these drugs to get through the approval process, and the success rate is very low. In fact, for oncology, less than 6% of drugs that enter clinical trials actually make it through FDA approval. And as we look at why drugs are failing, drugs that enter the clinic often fail right off the bat due to problems with safety in first-in-human trials. And if they do make it through phase 1 and are safe in healthy patients, they often fail in phase 2 due to efficacy. So this really led us to the hypothesis that if all of these drugs are going into clinical trials because they've worked in our preclinical models, then our preclinical disease models must be failing, and that there has to be better models that can better predict the safety of efficacy of drugs before going into human patients. When we look at various types of preclinical models, we found that the pig is amazingly anatomically and physiologically similar to humans by so many different comparisons. So if you look at the size of the pig, the anatomy, the respiratory rate, the blood volume, and various organ sizes, composition, and function, the pig is remarkably similar to the human patient. For me particularly, I've always been very interested in diseases of the brain, including brain tumors and neurodegenerative diseases. And when I just take a look at the human brain compared to the mouse brain by overall size, you can see the drastic difference in size. When we actually blow up the mouse brain to be the same size as the human brain and look at the structure, you can see how vastly different the structure is between the mouse and the human brain. But when you look at the pig brain compared to the human brain, you can see that they're very similar in both size, structure, and function as well. So the pig has these striking similarities in anatomical structure, physiological function, as well as genetic makeup to the humans, making an ideal preclinical model to test new therapeutics before going into the clinic. The pig has also been used for many decades in preclinical research for safety studies in medical devices. And at Recombinetics, we've really pioneered the ability to gene edit the pig. So we can now efficiently gene edit the pig to mimic human disease, which gives us the opportunity to address a broad range of diseases of different organs and indications. So this is the pipeline of Surgeon and really what we focus on. We do a lot of work looking at human genetics and specific genes and even specific gene mutations responsible for human disease. We then engineer those changes into the pig genome, and this is showing a TALEN, but we also use CRISPRs, base editors, transposons, various gene editing tools to mimic these genetic changes in the pig genome, which then gives you a genetically engineered model that replicates human disease, both anatomically, physiologically, and genetically. And these pig models can now be used for both safety and efficacy testing preclinically to better determine whether a drug is or a device is going to be successful in clinical trials. I want to point out that in addition to using these pig models for drug and device development, we've also used these pig models to look at things like combination therapies, new indications for old drugs, surgical interventions, biomarker development, the development of novel imaging technology, and others have used these pigs for learning and behavioral studies, which is pretty unique to the pig, as their learning and behavior is very similar to human. So at Surgeon, we, over the past 12 years, have developed a collection of swine biomedical models. In the early days, we were very focused in the cardiovascular space and medical device development and developed many models around cardiovascular diseases. More recently, we've been focused on developing models that can be applied towards imaging and novel therapies like gene therapy and cell therapy. And so you can see many of the different disease models that we've developed on this slide. Today, I'm going to be focused on our cancer models, but would be excited to tell you about these other disease models that we've developed in the future. So this is an overview of our oncology program, and I'll be telling you about 4 different parts of our oncology program today. I'll start by telling you how Surgeon has used germline editing to create Minipig models predisposed to cancer development. I'll then tell you about our new platform where we use somatic cell editing to induce cancer somatically. And then I'll finish by telling you about how we've used gene editing to create immunodeficient pigs and even taken that a step further to humanize these pigs for immuno-oncology applications. So I'm going to start by telling you a case study about our pig model of neurofibromatosis type 1. Neurofibromatosis type 1, or NF1, is a rare pediatric disease with very few therapeutic options. 1 in 3,000 children born will have NF1, and 50% of these will have inherited it from a parent, while 50% will have de novo mutations in the NF1 gene, being the first in their family to have neurofibromatosis type 1. There are many clinical signs of NF1, including dermal changes that occur at birth and throughout puberty. These are shown here on the right, where you can see cafe au lait macules or hyperpigmentation of the skin, which is diagnostic of NF1. Almost all NF1 patients also develop neurofibromas. So these are tumors that can be disfiguring and very painful to patients. Many NF1 patients develop skeletal abnormalities and scoliosis. 50% of the patients are affected by learning disabilities. And one of the most challenging and difficult parts of this disease is that these patients are predisposed to cancer. So 10% of NF1 patients will develop a malignant peripheral nerve sheath tumor. This is shown right here. These are soft tissue sarcomas that are very devastating. The 5-year survival rate is less than 50%. And sadly, 1 in 5 NF1 patients will develop a brain tumor. And often these are optic pathway gliomas or astrocytomas of the brain. And most often these occur in pediatric patients under the age of 7. These optic pathway gliomas can cause vision loss, and astrocytomas and other brain tumors can cause cognitive impairments. There's no cure for NF1, and new therapies are desperately needed. So the symptoms generally are managed. Patients will undergo treatment for nerve pain and therapy for learning disabilities. Problematic neurofibromas and optic gliomas can be surgically resected. And recently, actually in April this year, Selumetinib became the first FDA-approved therapy in NF1, but right now Selumetinib is only approved for inoperable plexiform neurofibromas that occur in these patients. The malignant peripheral nerve sheath tumors, or MPNSTs, that I showed you on the last slide have a very low 5-year survival rate, shown here on the graph on the right, of less than 50%. These are treated generally by radiation and or chemotherapy, and they're very difficult to surgically resect, oftentimes requiring an entire limb to be removed in order to get clean margins on a MPNST. So in order to address many of the challenges in NF1, a lot of mouse models have been developed over the years. And you can see in this table some of the mouse models that have been developed. And these mouse models really have given us a better understanding of the biological basis for this disease and started giving us some insights on therapies, but many challenges still remain. For example, we don't fully understand the molecular mechanism by which tumors develop in NF1 patients. Primary cell lines have been very difficult to generate from both mice and human patients. It's unclear which cell type needs to be targeted with therapies. So for example, in these neurofibromas, we know that the initiating cell is the Schwann cell. However, neurofibromas are packed with other cells, including fibroblasts and mast cells and other immune cells. The tumor biology still remains elusive and we don't fully understand it. There's a large gap in knowledge between genotype and phenotype relationships, meaning that 2 patients that have the exact same mutation in the NF1 gene often have very different phenotypes. And therapeutic development has remained challenging. So I mentioned the FDA approval of Selumetinib, but despite a lot of work in this disease over the past 3 decades, that's the only FDA approval that has come. And I do want to point out how successful Selumetinib has been for NF1 patients. This was a drug developed by AstraZeneca that I mentioned became the first FDA-approved therapy this year for inoperable plexiform neurofibromas. And you can see these 2 patients before and after that were treated with Selumetinib. Their plexiform neurofibromas shrank significantly. However, despite the approval of Selumetinib, there are still therapeutic challenges that remain. There are many other MEK inhibitors, which is the class of drug that Selumetinib is, under investigation, and there's considerations for combination therapies. And we really need better preclinical models to understand which of those drugs or which of those combinations is going to best treat our patients. There are new classes of drugs being considered for NF1 patients that need to go through preclinical evaluation before going into the clinic. There's a high bar for safety, efficacy, PK, and PD for all of these therapies that really needs to be understood before drugs can go into the clinic. Any drug for NF1 related brain abnormalities has to cross the blood-brain barrier, and this can often be difficult to assess in rodent models. There's some question on whether drugs can be used in the NF1 patient population prophylactically to prevent tumors from developing. And there's also consideration on using drugs to shrink tumors pre-surgically that should be preclinically evaluated. So with all these biological and therapeutic challenges, we set out to revolutionize preclinical research by making a human-sized preclinical model of NF1 that displays all the clinical features of NF1 that a lot of the mouse models are missing. So for example, none of the mouse models display cafe-au-lait macules. A lot of the dermal changes have been very difficult to model in the mouse. And mice don't undergo the same molecular mechanism of disease of human patients. So we took a mini pig and engineered in a human-specific NF1 mutation. And the first thing we found in these pigs is that all of the pigs develop these cafe-au-lait macules that almost every NF1 patient develops, but have never been modeled in the mouse. Our pigs meet the NIH diagnostic criteria for NF1 based on these cafe-au-lait macules alone. And when you look at them histologically, they are nearly identical to human cafe-au-lait macules. Our pigs also develop optic pathway gliomas and cutaneous neurofibromas, which are two of the most common tumor types in NF1 patients, and have been both very challenging to model in other animal species, including the mouse. When we isolate Schwann cells from our cutaneous neurofibromas or melanocytes from our cafe-au-lait macules, which are the initiating cells for these two phenotypes, we find loss of heterozygosity of the wild type NF1 allele and complete loss of neurofibromin protein. This is the exact same mechanism of disease that happens in human patients, and this has never been modeled in the mouse before. So in all those mouse models that I showed you, you had to use the Krelock system to biallelically knock out NF1 in the cell, the initiating cell of your choice. But in the pig, just like in human patients, they undergo this molecular mechanism spontaneously. These pigs also show myelin decompaction by electron microscopy, another phenotype that hasn't been seen in mice. And there are many applications that the mini pig is great for that are challenging to do in smaller animals. For example, we can do longitudinal blood and tissue sampling. We and others have done behavioral and learning studies. And today I'll tell you about some of the pharmacology and toxicology studies that have been possible in this model. So one of the first studies that we did with our NF1 mini pigs was to look at whether the pharmacokinetics of a drug in the mini pig were predictive of what happens in human patients. This has been one aspect of mouse models that has been really challenging because mice are so small, it's hard to get enough blood or plasma to really assess precisely the pharmacokinetic parameters. And when those types of studies have been done, the mouse has not always been predictive of the pharmacokinetics of a drug that you see in human patients. So here on the left, you can see in the mini pig, the plasma concentration of Selumetinib over time after a single dose of drug was given orally. And on the right, you can see the human plasma concentration over time of a single dose of Selumetinib given orally. And you can see that in both cases, the plasma concentration over time mimics each other very nicely, showing that the mini pig is predictive of human pharmacokinetics of this drug. We next wanted to ask, can this drug reach various tissues in the NF1 mini pig? So we all know that Selumetinib has been FDA approved for inoperable plexiform neurofibromas. So clearly this drug is able to reach deep nerves from which these plexiform neurofibromas arise, like the sciatic nerve. But what this study showed is that after a single oral dose of Selumetinib, we were able to get detectable drug levels in the cerebral cortex, the optic nerve, and the skin. And in fact, in all three of these tissues, the drug concentration was higher than what you see in the sciatic nerve, suggesting that Selumetinib may be effective at treating various glial tumors like astrocytomas, optic pathway gliomas that arise from the optic nerve, and cutaneous neurofibromas in the skin. We then wanted to ask if the drug can reach these different tissues, can it actually hit its target? So Selumetinib is a MAP kinase or MEK inhibitor. And so to read out whether this drug is hitting its target, we look at the expression of phospho-ERK. If Selumetinib is working, you'll see that phospho-ERK expression will be reduced. So when we look at the skin, we can see that with no drug in either the wild type or the NF1 pigs, Selumetinib is able to reduce phospho-ERK signaling, showing that it's hitting its target. We also see the same thing in sciatic nerve, where both wild type and NF1 pigs have a high level of phospho-ERK in the sciatic nerve, which is reduced with Selumetinib treatment. Interestingly, in the brain, in both optic nerve and cerebral cortex, only the NF1 mini pigs actually have an increase in phospho-ERK in these tissues. And in both cases, Selumetinib normalizes phospho-ERK levels to wild type levels, suggesting that Selumetinib is likely to be very effective at treating central nervous system abnormalities in NF1 patients. So this and other research that we've completed really has revolutionized preclinical research in the NF1 field. The NF1 pig has become the ideal model for assessing both pharmacokinetics and pharmacodynamics. It's exciting that a drug like Selumetinib shows similar pharmacokinetics in the pig as well as the human patient, but also that a small molecule drug that's a kinase inhibitor directed at a human kinase is also able to target a pig kinase and perform the same function by reducing that kinase signaling through reducing phospho-ERK. This model has been able to show us how well various drugs cross the blood-brain barrier. We're able to assess genotype-specific effects in various tissues with various drugs. We can really understand how well a drug targets specific tissues. And the exciting part about these experiments is they really can't be done in mice because you can't get enough tissue from mice to accurately assess how much drug is in that tissue. And there are certain regions that you just can't biopsy in a human patient, like brain regions and nerves. And these are all studies that can be done in the NF1 mini pig that are very critical preclinical studies. The mini pig is able to be used for dosing strategies, looking at safety and toxicity, efficacy, expanding indications, and combination therapies. And these are all things that we currently have ongoing and are very excited about the use of the mini pig for these different aspects of preclinical research. So I will mention that in addition to NF1, we're also developing many other mini pig models through germline editing that are predisposed to cancer development. We have a Neurofibromatosis Type 2 pig, and we're currently developing a tuberous sclerosis pig as well. But I'm going to move now onto our somatic cell gene editing platform to show you how, in addition to doing germline editing, we can also do somatic cell editing to induce cancer in our mini pig models. So our somatic cell gene delivery method is really a robust platform for tumor induction. We can do cancer induction by genetic alterations in somatic cells that confer a proliferation and survival advantage to these cells in the exact same way that human patients develop cancer with mutations in somatic cells and accumulation of mutation in somatic cells over time. This really offers us a flexible on-demand selection of what combination of oncogenes and tumor suppressor gene modifications we want to include in our tumor models, and allows us to be very flexible and adaptable as we develop these models. So as I tell you about this, we've really focused this platform on developing glioblastoma and hepatocellular carcinoma, but are now looking and excited about expanding this platform to other cancer types as well. So this is all done by direct injection of gene editing and gene modification tools into the pig. We can deliver transposons for stable gene delivery. So in this way, we can overexpress oncogenes, we can express dominant negative tumor suppressor genes, and we can express reporter genes. We also deliver site-specific nucleases. This can include CRISPRs, TALENs, zinc finger nucleases, any type of site-specific nuclease you could imagine. And that allows us to knock out tumor suppressor genes in the endogenous swine tumor suppressor genes. And everything that we deliver in this way, we deliver on plasmid DNA. And so we can deliver plasmid DNA for transient gene expression to express transposase that'll mobilize transposons to allow for stable gene expression. We can deliver things like fluorescent proteins to tag targeted cells, et cetera. So why did we focus on glioblastoma? Glioblastoma is a disease that has a desperate need for new therapies. It's the most common and aggressive brain tumor with 23,000 new cases per year in the US. The 5-year survival rate is extremely dismal for glioblastoma of less than 3%. And the standard approved therapies are surgery, radiation, and chemotherapy. And this standard of care for GBM has not changed for many, many years. GBM, as this has been studied over the past few years, it's become really clear that there are different subtypes of GBM with various pathways and genetic mutations. And it really points towards the need for tailored therapies. But right now, the standard of care for any patient with GBM remains the same, with surgery, radiation, and chemotherapy, and no targeted therapies currently approved. So what are out there for GBM models currently? A lot of what we know from GBM comes from rat and mouse models, which are very useful for the studies on the mechanism of GBM development, but less so for testing new therapies. Surgery is the first-line treatment for GBM, and this is hard or impossible to do in mice and rats. Therefore, if you really want to mimic or really want to study a new therapy against the current standard of care, that's almost impossible to do in rodent models. Fortunately, there's a model in dogs that develop GBM-like tumors, but this is a spontaneous model, so it's pretty rare, and it makes enrolling patient dogs very challenging to get enough dogs to do a study for GBM. And when groups have looked at the genetic similarity between canine and human tumors, they see that they vary quite a bit. So these dog tumors may not genetically represent human tumors as well. So we've developed a powerful new GBM pig model to accelerate the development of new therapies. And this is the first engineered immune competent large animal of GBM that's out there. So our approach, like I mentioned in the previous slide, is direct injection of gene editing tools and reporters into the brain for gene delivery to cells of the subventricular zone to induce GBM. And this is the first time this has ever been achieved in a large animal. So the advantages of this approach is that it's very efficient. We can induce tumors starting at birth, and these tumors develop very rapidly. And it's also very adaptable. So we can change the genes we target on demand. We can use human genes or make specific gene mutations that a drug targets. And it allows us to put together gene combinations to model very specific brain tumor subtypes. And again, on the right here is just a reminder of how similar the pig is to the human brain that you don't find in both the rat or the mouse, or even the monkey. So this is our Kaplan-Meier curve of our GBM brain tumors, and you can see that these tumors develop quite rapidly and the animals succumb to disease quite rapidly. There's a subset of animals that die of their GBMs by about 2 months, and another subset of animals that die between 4 and 5 months of age. This does exclude some animals that were imaged by CT at specific time points, but these were all animals that actually died of their brain tumors in this Kaplan-Meier curve. So like I mentioned, there's sort of two cohorts of animals, the ones that die early and the ones that die a little bit later. On the right here are brain tumors from 2 animals that died from disease by 2 months of age, and you can see just how large these brain tumors are. This one, for example, takes up a large volume of the hemisphere, and this one actually crossed into the other hemisphere as well. The histology analysis on these tumors is underway, but so far they, by histological analysis, resemble human GBM. And these tumors are able to be imaged by CT scanning, which is really exciting. Because most brain tumors are imaged by MRI, so the flexibility to use CT scanning to image these tumors allows a lot more flexibility in setting up preclinical studies. This tumor you can see in a coronal view here and a sagittal view here, and this is the actual gross tumor here. This is in a 3-month-old mini pig and was detected by CT 75 seconds post-contrast. These tumors develop in multiple regions throughout the brain. So the injection of the gene editing tools is directly into the lateral ventricle, as I mentioned, to target the subventricular zone. And about 50% of the tumors that we're seeing actually arise in the lateral ventricle. About 37.5% of tumors develop in the frontal lobe, and you can see these 2 tumors here that developed in the frontal lobe. And then we did have 1 pig that developed a tumor in the corpus callosum. When we look at the genetic modifications in these tumors, we see that the majority of tumors have mutations in NF1, P10, P53, ATRX, and RB1. These are the 5 tumor suppressor genes that we targeted with CRISPR. And in addition, we can detect stable transposon integration of many oncogenes that we also delivered on plasmid DNA using transposons and transposase. So we can see expression of EGFR, MET, NRAS, PDGF-beta, and AKT. So in summary, we've developed a platform that induces rapid genetically defined brain tumors in pigs. These brain tumors develop quite quickly, some in less than 2 months at a very high penetrance, in a brain that's much more similar to the human brain in both size and anatomy. This is very early in development, and so we're still working on this platform, and our next steps are to really characterize tumor cell invasion into the surrounding brain, and we already have evidence that these tumors are highly invasive and have evidence of gene editing in tumor cells in other regions of the brain outside of the gross tumor. We're excited about our next steps in applying this platform to model different GBM subtypes, and those are listed on the right here. The different GBM subtypes are pretty well defined, and we know the mutations associated with those. So we're really excited to, now that we've shown this proof of principle that we can develop brain tumors, to go down the path of modeling each of these subtypes and seeing whether or not they mimic the human brain tumor subtypes. And then we're now applying this model for preclinical therapeutic evaluation. So the human-sized model really allows us to do therapeutic efficacy and tox studies and compare new therapies to the current standard of care in glioblastoma. The fact that these pigs have a full immune system will allow us to test immunotherapies and do tumor microenvironment studies, which cannot be done in immune-suppressed animals or xenograft studies. And we're excited about the ability to develop new imaging modalities, surgical techniques, look at things like local drug delivery and medical devices, and more in this model. So now I'll switch gears a little bit to tell you about our hepatocellular carcinoma model. And similar to GBM, there's a desperate need for new therapies in liver cancer. Hepatocellular carcinoma is the most rapidly increasing cancer death rate in the United States. It's currently the 4th leading cause of death from cancer worldwide. And the overall mortality is 95%. And the 5-year relative survival of HCC is really dependent on what stage of this disease is diagnosed at. So local disease, you have a better chance of survival. But when the disease has spread either regionally or distantly, the survival goes down dramatically. And unfortunately, for most patients diagnosed with HCC, they're diagnosed at later stages of disease. And so our approach to modeling liver tumors in the Minipig is to do hydrodynamic injection of gene editing tools and reporters directly into the liver of neonatal mini pigs. We do this by using a catheter system where we use either a single or double balloon catheter threaded right into the umbilical vein of a neonatal mini pig. We can use a single balloon catheter to target the entire liver by hydrodynamic injection, or a double balloon catheter to just target the left lobe. So this is very much less invasive than other surgical methods that have been developed for large animal hydrodynamic delivery. The production of this model doesn't rely on fluoroscopy or advanced imaging. We induce tumors starting at birth, and these tumors develop quite rapidly. And we're also developing a reporter gene so that we can have an analysis of tumor development without the need for advanced imaging. So this would be a simple blood test that would tell us the volume of tumor in any given animal at any given time. Similar to GBM, our liver tumor platform is very adaptable, so we can change the genes we target on demand. Again, we can use human genes or make specific gene mutations that a drug targets. It allows us to do various gene combinations. And I think one of the really exciting developments in this model is that by being able to target the entire liver or just the left lobe gives us a lot of flexibility. So I'll show you a picture right now of the different lobe targeting. So when we just inject a GFP plasmid and do the full liver hydrodynamic injection, we can see that many cells in both the left lateral lobe and the right lateral lobe are targeted. But by using the double balloon catheter system, we can just target the left liver lobe, leaving the right liver lobe unedited and completely normal, which gives us the flexibility if we're testing a new therapeutic to test both how it does in a diseased liver as well as a healthy liver in the same animal. So this study is still ongoing, but I'll just show you a sneak peek at some of the early data. So we necropsied 2 animals that were just under 4 months old, a male animal and a female animal. We picked both sexes because, as you all know, HCC shows up very differently depending on whether you're male or female. In the male pigs, we found 6 liver tumors ranging in size from 1 to 5 millimeters. So pretty small tumors, but there were several of them at this early time point. And in the female pig, we found 5 liver tumors ranging from 2 to 4 millimeters. Again, pretty small, but exciting developments, and the full histological analysis is underway. But we believe that these tumors resemble HCC, and as this model progresses, will progress to full-blown HCC. So again, we're really excited because we think that, or we know that the pig liver is very similar to the human liver in many different ways: the size, the function, the segmental anatomy, the biliary tree, the hepatic vasculature, very, very similar between pig and human. Excitingly, the pig is more prone to fibrosis than mice, and in human patients, often liver disease underlies liver cancer. So fibrosis is a very important aspect of this model. We will be able to develop other underlying diseases in the mini pig that can be layered on as comorbidities to our HCC model. So for example, alcoholic liver disease from feeding ethanol on the bottom right here shows some other liver diseases that we either are going down the path for developing, such as NASH, or could easily develop by expressing viral proteins like HBV and HCV. We've also got a model that's starting to develop fibrosis. So the ability to have a preclinical model that can have underlying liver disease in addition to hepatocellular carcinoma is really critical for understanding how well therapeutics are going to work. So in summary of our liver cancer model, we have a platform, again, that induces rapid genetically defined liver tumors in pigs. These liver tumors develop very quickly within just a few months. And so far, we've seen very high penetrance. Every pig we've looked at has had liver tumors. We're going down the path of characterizing fully these tumors for penetrance, latency, and targeted genetic changes. And then similar to GBM, applying the platform to model different human HCC subtypes. And you can see those defined molecular subtypes up here on the right. And we look forward to modeling each of those subtypes and looking at the similarity to the human HCC subtypes. And again, like GBM, we're also applying this model to preclinical therapeutic evaluation. So having a human-sized model really allows us to do those therapeutic efficacy and tox studies, compare it to the standard of care, looking at immunotherapies and tumor microenvironment, and again, surgery, ablation, embolization, local drug delivery, all of these things that require a human-sized preclinical model will be feasible in this model. So switching gears once more, I'm going to finish this up by telling you a bit about our immunodeficient pig platform that we're developing that is a great model for cancer xenografts, and then how we're humanizing that pig for immuno-oncology applications. So we've been able to gene edit pigs to be immunodeficient. So these pigs have 2 mutations that are known to cause immunodeficiency. And these knockout pigs lack T cells, B cells, and NK cells, and that's just shown on the flow cytometry chart on the right, where you can see the wild type pigs have a population of B cells and a population of T cells, and our knockout pigs do not have those populations of cells. I didn't show the NK cells, but these pigs are also knocked out for NK cells. This is an exciting model because it doesn't require immunosuppressive regimens, and by every test that we've done so far, these pigs are much more immunodeficient than any immunosuppressed pig on drugs has ever been shown to be. With opening up this niche of immune cells, it opens up a niche that allows us to humanize these pigs, and I'll tell you a bit more about that in the next slide. But we think this large animal immunodeficient model is really exciting for xenograft studies and even orthotopic transplantation of xenografts. So you could implant human tumor cells in the exact location where a human tumor would develop to use these pigs as a preclinical model. They're also a great large animal for cell and gene therapy studies where the immune system or having a suppressed immune system is really important to study these types of therapies. They can, again, be used for safety and efficacy. They're not going to develop any anti-drug antibodies or things like that. A large animal is great for gene therapy scale-up studies, as well as understanding therapeutic delivery systems. All right, so moving on to the humanized platform, I told you in the previous slide about the immunodeficient pig. We really think that this pig will be a great bioreactor for human cells. So you can imagine that if you had a human cell or a human patient that was a cell donor, you could have stem cells that would go into this immunodeficient pig and actually create many of the cells that are difficult to create in vitro or to isolate from human patients themselves. And we're doing this right now, trying to fill this immunodeficient niche with human cells to make a humanized immune system in the pig. And we're doing this by several mechanisms. So this is our bioreactor for human cells. This is our immunodeficient pig. It lacks immune cells. We've been able to further engineer this pig to express human cytokines. We've eliminated phagocytosis of non-pig cells in the pig. And we've done additional modifications that allow us to put in human cells and actually have them grow and graft and function in the pig. We're now developing collection and processing systems to be able to take these human cells out of the pig. We're further engineering the human cells that go into the pig to potentially be therapeutic cells. And we're developing specific housing for these pigs to allow them to survive long-term for the engraftment and production of these human cells. So you can imagine two oncology applications for this bioreactor pig. One is that if we were to engineer these stem cells to have something like a CAR, you could use this for the production of human therapeutic cells like CAR T cells, CAR NK cells, where this pig itself would produce many, many therapeutic cells that could be used for adaptive cell therapy. The other application that we could imagine for these pigs is that having a swine with human immune system would allow for preclinical immunotherapy development and validation studies. And so we're also really excited about using this pig with a human immune system to look at various immunotherapies preclinically. And so with that, I just wanted to focus on the fact that this pig as a bioreactor would allow many, many different types of immune cells to be produced. And we think that in the lifespan of one pig, you could treat up to 270 patients without external cell expansion for various types of immune therapy cells. And the CAR T market alone is vastly growing, and there's a lot of development going on right now. So we're really excited to think about the pig as an alternative for in vitro off-the-shelf development of CAR T therapy. So I'll end here to talk a little bit about getting started and planning for pig models in your oncology studies. And there's lots of considerations to be made. There's clinical considerations. So I think pigs definitely deliver value where mice cannot. It's important to think about other large animal models, such as canine models. Canine models have been used a long time in diseases like glioblastoma, and they can be very good for certain studies, but they can also be challenging. We talked about how canine glioblastoma is very rare, so enrolling enough animals to do a preclinical study in dogs can be very challenging. Pigs versus non-human primates. Non-human primates are very expensive, they're difficult to produce, and there's not a whole lot of models in primates of human diseases, so pigs can be very good if you want to do efficacy studies and don't have a non-human primate that has that specific disease. I think it's important to think about when the pig is not great. So screening drugs is probably not something that you would want to do in a pig model, just because pigs are expensive and they are large and they are more difficult to work with versus mouse models or in vitro models. But if you really want precise safety, pharmacokinetic, and efficacy studies, I think the pig is a great model. Thinking about FDA requirements, there's a lot of medical devices that don't require efficacy models to go into the clinic. But if you look at the FDA's recommendation for these newer advanced therapies like cell and gene therapy, there's definitely a recommendation to use a large animal disease model in your research, and the pig could be ideal just for that. There are definitely challenges in clinical trials, especially with rare and orphan diseases. And this is why we were really excited about the pig model of NF1, because when you have a rare disease or a pediatric disease that's really hard to enroll enough patients to do multiple clinical trials every year, you need better preclinical models to identify which therapies you're going to move into the clinic. There's also some logistical considerations to think about with a pig. For example, how many animals you're going to do per study. We think that the amount of data that you can get from a pig will lower the number of animals that you need in any given study. So whereas you might need 20 or 30 mice, you may only need a half a dozen or a dozen pigs to do the same study and get the same statistical power out of it. We have to think about timing in the pigs, and all the disease models that we create, we really try to find a balance between modeling the natural history of disease in human patients, with also understanding that for preclinical development, we don't want to wait years for a model to get a tumor to develop. There's a lot of logistical considerations with housing and shipping. Luckily, we have our own internal housing for animals, but we also partner with a lot of CROs that allow housing of pigs and are really good at taking care of these pigs. We can ship pigs anywhere in the United States and have ways to get them outside of the United States as well. And then, of course, budget considerations. So with that, I will hand it back to Bill. That's the end of my presentation. So Bill?
Yes, thank you, Adrienne. I just wanted to remind everyone that you can put your questions into the Q&A. We reserved a little bit of time here at the end. And just before we start that, I just wanted to encourage everyone to just pause for a moment. It's kind of thought-provoking to step back and think of the implications of what was just presented. So Sarojin and Adrienne's team can genetically edit pigs to induce the ideal disease models for developing new drugs, testing safety, pharmacokinetics, and efficacy before all prior to human testing, and with unparalleled predictive capabilities of human response. So think back to that 97% failure rate of oncology drugs taken through the clinic. Think back to that 8 to 15 year development rate and transforming both of those numbers by orders of magnitude and leveraging platforms such as this to accomplish that, and what that's going to mean for the rate of making progress on these terrible diseases. First question that came in is, do you have lung and pancreatic cancer models in the pipeline?
Yeah, that's a great question. I think both of those models are front and center as we think about what our next models are going to be. I mean, clearly, there's definitely a need for preclinical models in the lung. I think that's been one of the types of cancer that's been very difficult to model in other animals. And I think pancreatic cancer, there's just a huge need for therapeutic development. And so we're actively thinking about the best way to do that. And we're looking for partners to be able to push that forward for preclinical pipelines of other companies as well.
Okay, thank you. And I edited out inadvertently. There was a preamble to that question, which was a very nice lecture. So, okay, we have another question. What types of pigs are you using and how big do they get?
Oh, that's a great question. So most of the data that I showed today was in the Ossaba Minipig. And these pigs get to be about 200 to 250 pounds. They get to be about the size of a human adult. The reason we chose the Ossaba pigs for a lot of our early work is because they actually develop metabolic disease over time. And so they've been a really nice model to study cardiovascular disease, comorbidities, underlying liver disease, things like that. But we also have access to many other types of mini pigs, including the Yucatan mini pig, which is a very popular preclinical model for preclinical studies, as well as the large white farm pigs. So depending on the type of study, the size of the animal, how big of litters you want, we could really have the opportunity to pick any type of pig, and we can engineer those for the specific model that you want to develop.
Okay, and again, if you're wanting to put in questions, use the little Q&A at the bottom of your Zoom window, and I'll field them up. Here's one that came in. How challenging are preclinical drug studies in pigs, and what routes of administration can you use to administer drugs to pigs?
Yeah, that's also a great question. So really any route of administration that you could imagine for a human patient can be used in the pig. So the Selumetinib study I showed was oral administration. We've been able to give drugs subcutaneously, intramuscularly, dermally, IV, inhalation, really any route of administration. And preclinical studies with drugs with pigs are actually fairly straightforward. So the size of the pig makes it pretty easy both for drug administration and sample collection. So the size of these pigs lets us take, if we're looking at the skin, we can take many biopsies. We can actually implant vascular access ports and take blood from these pigs every 10 seconds if we wanted to. So preclinical studies around both drugs and biomarkers are fairly straightforward to do in the pig just because their size makes it so easy to do all of these things.
Okay, great. And are there classes of therapies for which the pig is better suited as a preclinical model than others? For instance, for example, they ask, is a small molecule that won't hit its target in the pig?
Okay, so that was one of the questions I had when we first went down this route is, even if we develop the most ideal preclinical model that mimics human disease perfectly, are human drugs actually going to be able to go into the pig and act in the same way that they would in the human patient? And so the great thing about the pig is that the whole genome has been sequenced, and we can do a large bioinformatic analysis before we start any study. And we can really understand for specific proteins, do they have the same epitope something might be binding or something a drug might be targeting. When we've looked, especially with oncology drugs and kinase inhibitors and small molecules, it's amazing how similar the pig and the human are genetically, and that these drugs actually go in and they hit their target and they work in the same way as the pig. Selumetinib is a great example. This is a MEK inhibitor. It's a small molecule that goes in and inhibits MEK, which is a MAP kinase protein, and the drug does the exact same thing as the pig. And when we compare a lot of the cancer genes at the protein level, they're often 99% or 100% the exact same as the human protein. So I think there are many classes of drugs that are really exciting that can be studied in the pig and have that same translational value.
Okay, well, if there's no more questions, Adrian, I think that's a great place to leave it. And to thank you all for joining us. And we will be sending out a link to the recording that you can, if you came in late or had to leave early, you can review that. And thanks again for joining us this afternoon.
Thank you.